Good morning, one and all. Myself Indraja, Associate Professor of MRIPS under MRV. Today's our topic is amino acid metabolism. And what is amino acids? Before going to the inside of the amino acids, the proteins we have to know. The proteins are the linear molecules, polymers of the L-alpha amino acids. These are linked by covalent peptide bonds. And what are all the amino acids? Amino acids which contains, you can see the structure over there. It contains amino group. You can see this is the amino group and carboxylic acid group. So and then R R represents the groups and also side chain and H. So we call it as chiral carbon. The chiral carbon containing amino acids. Okay, and you can see that why we are calling it as alpha amino acids is because the carboxylic acid first carbon we call it as the first carbon, the second carbon is the alpha carbon, and then the third carbon is the beta carbon. So we are calling it as alpha amino acids. What are L alpha amino acids? The amino group is present at the L side, levo, left side. So we call it as alpha amino acids. Coming to the next slide, it is about ionized form of physiological pH. Coming to the next slide, it is about the ionized form at physiological pH, how it will be. These are the amino acids are of two forms, will be the L form and D form. The L form is the which contains the amino group on left side, whereas in the D form the amino group will be present at the right side. So you can see that amino group is there, carboxylic acid group is there, H and R is there. R is the side chain or the group will be attached to that. So we can call it as chiral center, which is optically active, and tetrahedral arrangement is there because of that four arrangements. Okay, and so we are call it as a stereoisomers and enantiomers. You can see the example also here, L glyceraldehyde and D glyceraldehyde, which contains a chiral carbon which is attached with the four different groups or atoms. Okay, so it is the L form, L alpha amino acid. Generally we call it as the amino acid as L alpha amino acid because of the chiral center and also enantiomers. Coming to the next slide, you can see the example of the lysine here, which contains a 6 carbon structure. The first carbon and the second carbon is the alpha, third carbon is the beta, and the fourth carbon is the gamma, and fifth delta and epsilon will be there, the sixth carbon. To the sixth carbon the amino group is attached. There are two amino groups are there. So normally the amino acids are of two types are there, standard amino acids and the non-standard amino acids. Our body contains more than about 300 amino acids will be there, but most important standard amino acids are 20 are there. Okay, these are essential for our body. And what are non-standard amino acids? Other than these 20 amino acids, we call it as D amino acids, non-protein amino acids, and amino acid derivatives. The derivatives of these 20 also we consider it as non-standard amino acids. Other than these 20 amino acids, 21 and 22 amino acids also will be there, like example selenocysteine, and 22 is the pyrrolysine. These are the structural example for the amino acid and also the classification of the amino acids. General classification, there are two types, standard amino acids and non-standard amino acids. Coming to the classification of the amino acids, there are four types are there. First one based on the structure and the chemical structure, and the second one based on the polarity and also the solubility, and based on metabolic fate and nutritional classification. These are the four types of classification of the amino acid. Based on coming to the first one, based on the structure and chemical nature, there are again divided into 7 types. That is, first one, aliphatic side chain. The side chain will be there for the amino acids in the amino acid basic structure. And the second one, the side chain with the OH group. The OH group will be present in the side chain, whereas third one is the side chain with the sulfur. The sulfur is present at the side chain, and the side chain with acidic group, and the side chain with the basic group, and aromatic, amino acids, and amino acids. This is the first classification based on the structure and chemical nature. Based on the polarity and also the solubility, there are two types will be there: hydrophilic and hydrophobic. Based on metabolic fate, glucogenic, ketogenic, and both. That means the amino acids involved in the glycolysis process and the ketone body's degradation process, and also both amino acids involved in glucogenic and ketogenic pathways. And coming to the fourth one, nutritional classification, which are essential, non-essential, semi-essential. What are all the amino acids essential for our body, which are not essential our body, and some of the semi-essential amino acids also will be present. These are the four types of classification of amino acids. Coming to the next slide. Based on the first one, structure and chemical nature, you can see that structure here. This is the basic structure. This is the basic structure of the amino acid. For this, it was divided into 5 types. In the first one, based on the aliphatic side chain, it is of glycine, alanine, valine, leucine, and isoleucine. Glycine. Coming to the first one, glycine, you can see the structure. The same, instead of R position, H is there. So it is not chirally active. That means optically inactive. It is not a chiral compound because different groups are present to the carbon atom. But here two H are there. Two H atoms are present at the two sides. So it is not chiral carbon and it is not optically active. Okay, the GLY is the short form of the glycine and we represent as G here. And coming to the second one, alanine, instead of R, CH3 is present, so we can call it as alanine. And coming to the third one, valine, you can see the side chain here. The side chain is present here, you can see that one. Two methyl groups are attached to the CH, and then leucine, the leucine, to the leucine, two methyl groups, and also 4 carbon structure you can see here. And then the side chain, you can see it is CH actually. This is the CH. So after this, again one more methyl group is added, so we call it as isoleucine. You can see the last 3 structures, these are chain form, structural form, open chain form. Okay, that is branched like it is there, so we call it as branched chain amino acids. In short form, we can remember it as GAVLI, GAVLI, that is glycine, alanine, valine, leucine, and isoleucine. Coming to the next classification, the side chains with the hydroxyl group. You can see instead of the CH, it is of 3 types in the side group: serine, threonine, tyrosine. You can see the structure. OH group is present at R position, R carbon, that is CH2, to that OH is attached. So we call it as serine. And threonine. In the threonine, you can see the same structure will be there. One more methyl group is added. We can call it as threonine. In short form, we are calling it as THR and T, whereas serine, SCR, S. Last one is the tyrosine. Tyrosine is T. We represent it as T by R or T. You can see there is a phenol structure is attached to the side chain. Then these are the side chain with the hydroxyl groups. These are the examples or the classification of the side chain with the hydroxyl group. Coming to the next one, it is a side chain with the sulfur atoms. Instead of OH groups before, it contains sulfur atom SH, you can see that one, and we can call it as cysteine. C by S is the short form and we represent as C. Methionine, MET, you can short form call it as M. To this, 4 carbon atoms are there and sulfur group is attached to it. We can call it as thioether also. So these are the 2 examples for the sulfur containing atoms, and one more is the cysteine. The 2 molecules of cysteine combined formed with the help of the sulfur hydroxyl group, then we can call it as cysteine. Coming to the next one, that is the side chain with the acidic groups or the amides or else acidic amino acids. These are our 4 types. You can see the first one, the aspartic acid, the second one is the aspargine, and third one is the glutamic acid, and the fourth one is glutamine. These are the structural examples. You can see the carboxylic acids are present. 2 carboxylic acids are present, groups are present at the carbon chain. So we can call it as beta carboxyl because the third carbon, which we call it as beta, to that carbon the carboxyl group is attached. So we call it as beta carboxyl here. This one is called as aspartic acid, whereas the aspargine, you can see the carboxyl group is present at the alpha position along with the amine group. Then we can call it as aspargine. And the glutamic acid, to this, these are the 4 carbon atoms are there. You can see to the 4th carbon it is present the carboxyl group, so we can call it as gamma carboxyl. And you can see the last one, glutamine, that is the carboxylic acid in the amino group is present for the 4th carbon. Here the carbon it was present. So this is the difference between these 2 carbon atom, these 2 amino acids. So these are the classification of the side chain with the acidic groups. Coming to the 5th one, the side chain with the basic groups, that is basic amino acids will be there. There are dibasic monocarboxylic acids also, we can call it as the basic amino acids. First one is the arginine, second one is the lysine, and the third one is the histidine. First one is the arginine. You can see the structure, the amino groups are present here. We can call it as these groups as guanido groups, whereas the lysine, the side chain to the side chain, you can see the same example we have discussed in the previous slide also, the lysine, which contains up to epsilon carbon, that is the amino group is present at the last carbon, the 6th carbon, then we can call it as epsilon amino groups. And then the histidine, which contains a 5-membered aromatic ring to the beta carbon. So we can call it as a imidazole. The imidazole ring is present at the 2nd carbon. Then it is called as imidazole group. Coming to the 6th classification, it is the aromatic amino acids will be present. That means the aromatic ring will be present at the amino acid in the side chain. These are the examples, that is the phenylalanine, tyrosine, and tryptophan. You can see the benzene ring is present in the first one, and the phenyl ring is present at the 3rd carbon. Then we can call it as phenylalanine. Whereas coming to the phenol structure will be present at the 3rd carbon, then tyrosine, and indole group is present at the 3rd carbon, then it is called as the tryptophan, and imido group will be present, then we can call it as proline. If imino group is imino aromatic ring will be present, then we can call it as proline. These are the classification for the aromatic amino acids. Coming to the 2nd classification, that is classification based on polarity or the solubility. There are 2 types will be there, amino acids. Amino acid 2 types will be there, hydrophilic amino acids and hydrophobic amino acids, which are polar and non-polar. Coming to the first one, the hydrophilic polar, First one is the negative charged and uncharged and the positive charged amino acids will be there. Are three types will be there. That is the hydrophilic. Under the hydrophilic amino acids, three types are there: negative charge, positive charge, and uncharged. Coming to the first one, the negative charge, there are two examples: aspartic acid and glutamic acid. And the positive charge, lysine, arginine, and histidine. Whereas coming to uncharged, without any charge, that is serine, threonine, tyrosine, asparagine, glutamine, cysteine, and glycine. Whereas coming to the hydrophobic, there are two types will be there: aliphatic and aromatic. In the aliphatic, alanine, valine, leucine, isoleucine will be there, and aromatic, phenylalanine and tryptophan will be there. If the amino group is present, then we can call it as proline and methionine. Coming to the third classification, based on the metabolic fate. As we discussed before, both glucogenic and ketogenic synthesis, some of the amino acids are involved. Those are phenylalanine, isoleucine, tryptophan, tyrosine. Only in the ketogenic pathway, that is the synthesis of ketone bodies, in that pathway leucine and lysine will be involved, and in glucogenic pathway the remaining 14 amino acids will be involved, and the examples are alanine, aspartate, glycine, methionine. Okay. All the 14 amino acids will be involved in the, that is in the 20 amino acid, the 14 amino acids will be involved in the glucogenic pathway. These are the examples and the classification of the classification based on the metabolic fate. Whereas coming to the fourth one, the last classification, it is based on the nutritional. These are three types will be there. First one is the essential amino acids, second one is the semi-essential amino acids, whereas third one is the non-essential amino acids. And the first one, these are the amino acids which are essential for our body, which are important for our body. Those are phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine, lysine. You can simply remember it as Praveit Tin Hall. Okay, you can remember it as Praveit Tin Hall. And coming to the second one, the semi-essential amino acids, it will be essential but not that much to our body. We can procure from the through the food also. And examples for that, histidine, arginine. And third one is the non-essential amino acids which are not essential. They are helpful in the reactions, other reactions, that is glycine, alanine, serine, cysteine, aspartate, asparagine, glutamate, tyrosine, and proline. Okay, these are the classification of the amino acids. For today's topic, I will end up to here. Thank you.